well, disappearing at certain moment of life that in male mice appear when it starts the puberty. Meanwhile, in female, it regress when they have the first pregnancy or in later stages of life, meaning like 20 months of life, but it's not really, it depends on the strain of the mice and everything. Moreover, we have the lacking of the CYP17A1 enzyme, leading also to, that has the consequence that there is no adrenal androgens produced in the mice, and beside that, the main glucocorticoid is the corticosteroid instead of the cortisol as in human. So in 2013, a team in Boston has first produced an aldosterone synthase cray mouse model in which the cray is under the control of the CYP11B2 gene that is coding for the aldosterone synthase. Their strategy was to introduce the cray recombinase in the exon 2, and meanwhile the exon from 3 to 9 were completely removed. That means that the homozygous cray mice lead to a CYP11B2 allele disruption of both allele and a loss of function CYP11B2 model that lead to the incapacity of the model to produce aldosterone and an overactivation of the renin due to the lacking of aldosterone production. So what they have done, they have used only the cray wild type mice, of course. That seems to be well, fine with also only an allele expressing the aldosterone synthase, the CYP11B2. This mouse model has been crossed with a classical MTMG mouse model in order to be able to do cell lineage tracing study. Just to explain a little bit, without the CYP11B2 expression, we don't have the cray expression, meaning that the cells of the entire body, actually, they are tomato positive at the membrane level. When there is the CYP11B2 expression, we have the cleavage by the cray of this sequence leading to the GFP expression at the membrane level. Besides that, it's important to remind that not only the cells that at least once express the CYP11B2, they become GFP positive, but also all the cells that are originated from those cells. And that was really important for the lineage tracing study that they have performed, in which they have demonstrated that the main pathway for the tissue renewal, for the adrenal cortex renewal, is the transdifferentiation of differentiated zona glomerulosa cells that are located here into zona fasciculata cells, and in the meanwhile there is centripetal migration along the cortex until, over time, as you can observe, reaching an entire adrenal cortex GFP positive, meaning that all those cells were at least originated from the outer layer that is the zona glomerulosa. These findings, anyway, does not exclude that the other pathways that were theorized at the time, for example, the zonal model, the subcapsular progenitor differentiation into the zona fasciculata directly, X-zone expansion, and self-duplication of the layer are maybe pathway that can be possible to have when we are in conditions other than the physiological ones. So to sum up a little bit, the adrenal cortex renewal is characterized by two main steps. We have the differentiation of the stem cell progenitor located beneath the capsule here and spread along the zona glomerulosa that become zona glomerulosa cells. This step is balanced and controlled by the interaction between the SHH pathway that is expressed by the stem progenitor cells and the Wnt beta-catenin pathway. There is a pathway mainly only expressed in the zona glomerulosa cells and that is promoting the zona glomerulosa cell identity. The second one, that is the one that I just described, is the lineage conversion of zona glomerulosa cells into zona fasciculata cells. That is the so-called transdifferentiation and their centripetal migration until reaching the corticomedullary boundary that is present here below, where they go through apoptosis. This second step is controlled by the interaction and mutual inhibition between the Wnt beta-catenin pathway and the PKA pathway. The PKA pathway is expressed by the zona fasciculata cells, so this layer. There is a sexual dimorphic dynamism between these two, the adrenal cortex renewal in the timing of happening of this phenomenon. We have the female mice that totally renew the entire adrenal cortex in 3 months. Meanwhile, male mice, they totally renew the adrenal cortex after 9 months, so a 3 times faster process in female mice. To go back a little bit on primary aldosteronism genetic causes, we have genetic mutation that can be somatic or germline mutation leading to the familiar form that mostly interest the ion channels and pumps, which may lead to a direct or indirect increase in cytosolic calcium concentration, which activate the calcium signaling and lead to the expression of the CYP11B2 gene and aldosterone production. There are some other mechanisms like chimeric gene or mutation on the beta-catenin gene and others, but mainly it happens in the ion channel and pumps. The lab has recently published a genome-wide association study where they have identified different risk loci for this disease. They identified mainly 2 genes, KZ1 and RXFP2. Interestingly, these 2 genes, in vitro study overexpressing these 2 genes in adrenocortical cells show that there is a suppression in the basal and stimulated mineralocorticoid output, in the aldosterone production mostly. And the current working model suggests that the pathogenic outcome may be genetically driven by the reduction of the aldosterone production by the zona glomerulosa cells when there are the expression of those genes, and that the lifelong increase stimulation of the zona glomerulosa cells in order to maintain an appropriate aldosterone production may create a propitious environment through an increase of proliferation to the appearance of somatic mutation and lately of the appearance of the primary aldosteronism. So the hypothesis of my project is that the genetic predisposition may be modulated by external factors such as dietary salt intake and stress, which may disrupt the adrenal cortex homeostasis and contribute to the development of primary aldosteronism. And this hypothesis, we have carried on with this hypothesis working on a new CYP11B2 model where the aldosterone production and regulation is conserved, more or less. So the new strategy that we have used is that instead of entering the credit combinase as before in the exon 2 disrupting the other exons, we enter the credit combinase in the 3' untranslated region of the CYP11B2 gene after an ERES region. This will allow the production of 2 different proteins from a single messenger RNA. We have analyzed the aldosterone plasma concentration of Cre mice, Cre wild type, and wild type, Cre wild type, and Cre mice, and male and female. All this experiment, due to all the differences that we have between the 2 sexes, has been carried on in both male and female mice. We observed that aldosterone doesn't change and is not significantly different in the 3 genotype of the mice. To better characterize the model, we have performed Western blot analysis, and we observed that even though it's not significant, we have a decrease in the homozygote Cre mice and a significant decrease also in female and a slight decrease in the Cre wild type. This decrease that we observe mainly in the homozygote mice doing immunofluorescence study of the aldosterone synthase show that there is a slight increase, a tendency in the Cre mice showing an increased percentage of cells expressing the aldosterone synthase. So actually our idea is that even though the total amount of protein is lower, there are more cells that are expressing this, the aldosterone synthase enzyme. We have no data about the capacity of the enzyme to produce aldosterone, so what we think is that maybe even a little amount in in a higher percentage of cells is enough to compensate and lead to a proper aldosterone production. Anyway, we also choose finally to use a Creuel type model in order to avoid any compensation process that is involved maybe. We have crossed also this new CYP11B2 CRE model with a reporter mouse model in order to be able to do lineage tracing study and to track that the expression of the CRE is tissue specific in the organ that we want to study, so the adrenal cortex, and not present in any other tissue studied. So in our model, studying the mice, male and female mice between 2 and 9 weeks, what we observe is that there are actually no differences between male and female in the progression of this tissue, different from previously published data in the literature, and this may be due to the time point of our study, so all the other experiments were carried on in adult mice. Meanwhile, when we consider mice from 2 to 9 weeks, we have also process of like growth of the mice, puberty that can be involved in this process that has never been described. We have actually a significancy, a difference between male and female mice at 4 weeks. That is more or less the time in which the X zone disappear in mice, and the X zone would be tomato positive, meaning that we will have an increase in the percentage of GFP positive cells in the male mice. Interestingly, what we have firstly described that has never been proven by other study is that the X zone, even though in male mice it disappeared at around 4 or 5 weeks, in female mice it remains until later stages of life, if the females are not inbreeding, of course. And we have observed that there is a link between zona fasciculata and X zone cells that has been proven by the co-localization of GFP positive cells and 20 alpha HSD positive cells. 20 alpha HSD is a marker of the X zone, so meaning that we have a certain degree of lineage conversion between zona fasciculata and X zone, that before has been kind of stated that it was not going on, that it was not happening because of the fetal origin of this layer. Meanwhile, the zona glomerulosa and fasciculata, they are mature tissue. We have studied also the progression of the transdifferentiation of the X zone, and what we have observed is that we have not a constant progression of it over time, and the hypothesis behind it is that the GFP positive cells that transdifferentiate also in X zone cells were the one coming from the zona glomerulosa, meaning that most probably those cells are more prone to go through apoptosis than the others not coming from the above layers. To modulate the plasticity of the adrenal, what we have done is a modulation of the renin-angiotensin-aldosterone system through through low salt diet, leading to the activation of the pathway, and high salt diet leading to an inactivation. The scheme, the treatment has been performed from 4 to 6 weeks for 2 weeks of treatment with the food, with like low salt diet food and high salt diet food for the first cohort, and the second cohort has also carried on a recovery period where the mice came back on a standard diet. So to be quick, the treatment was effective both in male and female mice. We observed an increase in the CYP11B2 expression and an increase in aldosterone production due to the treatment that goes on to the normality after 1 week of recovery, meaning that 1 week of recovery is enough to get back to a almost normal situation. For the high salt diet, we see a trend in decrease in the CYP11B2 gene and a suppression of the aldosterone production in the time point of the treatment that goes back to normality after recovery. What we have done is the analysis. Is the analysis of the aldosterone synthase enzyme through immunofluorescence, and that confirmed the decrease in the increase in the aldosterone synthase expression in the treated low salt diet mice that goes back to normality after 1 week of recovery. At this moment, we started to wonder if this increase in aldosterone synthase was also associated with a thickening of the layer. and we perform immunofluorescence analysis of for the DAB2, there is a marker of the zona glomerulosa cells, showing that interestingly, even though it has been proven that it should increase the size of the zona glomerulosa, there were no changes in the thickening, neither in the time point 1 of the treatment or after the recovery. We have analyzed also the Wnt beta-catenin pathway, that is the main pathway of the zona glomerulosa promoting the identity of those cells, and we also confirm that there are no alteration of this pathway, meaning that this pathway is most probably above the renin-angiotensin-aldosterone system stimulation. But to better understand what was happening, we decided to perform spatial transcriptomic analysis, so we could identify with the specific cell markers 11 cell cluster, and well, for the zona glomerulosa, zona fasciculata, that are the transdifferentiating cell, we use a mix of the two cell markers. This is the map show the connection that there is between zona glomerulosa, progenitors, zona glomerulosa, zona fasciculata, and X zone. Most probably like showing also that there is a lineage, like the conversion of those cells even in the UMAP by the proximity of the cell clusters. This is an example of the VIMM slide. So we could we use four slides, and this is the disposition of the tissue. So we had 8 to 9 tissue in each square, and this is the different cell marker expressed in the different cell clusters. So to do, we did a differential expression gene analysis in male and female mice. I'm showing you only the male mice for time reason. So what is interesting, we observed that there is an important transcriptional changes in the zona glomerulosa, as we would expect, but more importantly, we observe a really huge transcriptional activation or changes in the zona fasciculata, both in the time point of the diet and also after the recovery, meaning an involvement of this layer after the treatment and in the recovery process that we wouldn't have expected, because the low-salt diet is meant to target only the zona fasciculata layer. But what is more interestingly is that controlling the percentage of different cell cluster, we observe that there is an activation of the progenitor cells in the zona glomerulosa and fasciculata with a decrease of the transdifferentiating cell cluster, most probably meaning that we have a direct differentiation of the progenitor cells going to the zona glomerulosa and also to the zona fasciculata. So that is one of the alternative pathways that were like theorized before. After the recovery, we go back to normality. Female mice, they also show a huge involvement of the zona fasciculata in the treatment, and a really smaller involvement, smaller than the male mice of the zona glomerulosa. But interestingly, the modulation of the lineage conversion of the different layers is modulated in a different way, showing a slowdown of the transdifferentiation and a faster differentiation. So the progenitors, they are supplying the zona glomerulosa, and they are blocking the transdifferentiation, so the progression of zona glomerulosa cells to zona fasciculata. Sorry, this was okay, I can say. So what was also interesting is that analyzing the upregulated gene in male and the downregulated gene in female, we observed that there are 38 genes that are were differentially regulated between the two sexes, and we are currently analyzing if those can be involved, and they are also regulated by the sexual hormones. To confirm also our findings on the different process of lineage conversion, we have for male mice, we have do a segmentation of the different layer of the cortex in different layer of a certain thickness, and what we have observed is that go after the zona glomerulosa layer, that is after the layer 3, we saw that there is a decrease in GFP positive cells. As I told you, if we have a direct differentiation of progenitor cells into zona fasciculata cells, those didn't express the CYP11B2 and will not become GFP positive. So this is actually most probably what we are observing. We are increasing the N of this study to confirm our findings, and we use the transdifferentiation of the X zone in order to show that there is a slowdown of the transdifferentiation rate also in the X zone, as we are observing between zona glomerulosa and fasciculata, that after goes back to normality or is, let's say, also released with a faster transdifferentiation after the recovery. For the so for the isol diet, we did exactly the same. So we proved that there is a decrease in aldosterone synthesis percentage of cells in the percentage of cells expressing aldosterone synthase. There are also in this case no changes in the thickness of the layer marked by visualizing this slide by the DAB2, but in the response for the in the differentially expressed gene, we observe also in this case a huge involvement of the zona fasciculata both in the time point 1 and in the time point 2, and really little involvement of the zona glomerulosa in a treatment that is meant to target that layer. The analysis of the different cell cluster composition observed that there is a slower rate of this differentiation with an increase in progenitor cells that after progress until the different layer once we are in the recovery part. And the female, they show almost the same with an higher progression of the transdifferentiation in the recovery part. This has been confirmed also by no changes in the GFP percentage of positive cells in the different layers, and the progression of the in the X zone show the release of the transdifferentiation in the recovery period. So to sum up, and it's the end, we have generated a new CYP11B2 Cray mouse model in which the gene is not disrupted. We firstly described the transdifferentiation of the X zone layer, showing a connection between zona glomerulosa and fasciculata. The high salt and low salt diet modulate the zona glomerulosa as expected, but also show a huge involvement of the zona fasciculata, and this meaning firstly not firstly describe the interaction between these two layers in these pathophysiological challenges that we have performed. The low salt diet in male mice activate a direct process of differentiation of the progenitor cells into zona fasciculata, and meanwhile in female just a slowdown of the rate, as is also observed for the isol diet. So the perspective is to further investigate interaction, to maybe build better protocols for the diet, and would be perfect to have an inducible model for this study in order to skip the puberty part and don't have these confundances. And well, thank you for your attention.
Thank you very much, Nicola.
You're welcome.
Really different from what we usually do in the lab. Yes. Questions? Slightly. Yeah, there is a lot of microscopy.
Yeah.
Questions?
So the mitochondria, they are a really important, interesting part of this tissue, and as I think already I told you, so the study of the mitochondria in this tissue has been performed, I would say, in the 70s and 80s, and there were major discovery, and after it just stopped, and no one is doing anything about mitochondria. Yeah, no, but that is interesting because in the zona glomerulosa and the zona fasciculata, the network of the mitochondria are really different. So the zona glomerulosa, they have round-shaped mitochondria, crystal density is really high, and meanwhile in the zona fasciculata, they are elongated, the crystal density is decreased, but it's interesting this part because those are actually the same cell, because you have the switch from one kind of cell to the other one, meaning that what is going on in the network pool during this process, and most probably is an important process that is involved in the transdifferentiation and in the regulation. So summoylation, I think, that is involved in the regulation of the DRP1, and it has been shown that there is a gradient along the cortex of the summoylation process with that higher rate in the zona glomerulosa and decreasing until reaching the corticomedullary boundary. So I would say that there is for sure something, but no one is doing it.
Cells have this triangular
Yeah.
Yeah, so they really have crystal that looks like triangles, like this.
Yeah, but exactly. But also, I mean, I would say that all the steroid hormones production is happening actually at the mitochondrial level. So the aldosterone synthase and the 11 beta hydroxylase, they are located at the inner mitochondrial membrane. So you know you have the import of the core.
You know that you should read papers from the lab, right? Because you know that we publish on this. And if you don't know, you have to go back and read. Okay? Okay. It was Vasilevsky et al. And it was not in the 80s.
But
It was not even in the 90s. I was in medical school in the 90s. Okay?
there was an important professor from Padua that did a lot of study on the adrenal. That is Nusseldorf. Nussdorf. That it was one of the.
My professor of anatomy.
Okay. He published quite a lot about mitochondria in the adrenal, but after nobody followed it.
Yeah, because it was a pain in the ass.
No, but you say it's very interesting because actually the synthesis of the hormones it occurs at the contact sites between the mitochondria, ER, even peroxisome site.
The first step and the rate limiting one
is this
fully the inner mitochondria.
No, yes, but the synthesis of
The rest of
steroid
the-
hormones is huge. You have like, and if you go to the ER, it follows a
The
path.
synthesis of pregnenolone, which is the rate limiting step of the biosynthesis of steroid hormones, happens at the level of the inner mitochondria.
Yes, but
Okay.
all the other steps.
Then this thing is being transported somehow, okay, which is totally unknown, to mostly contact sites because you need to hydroxylate for this. So this happens outside of the mind. But there is it's super cool because first of all you need to get cholesterol in the inner mitochondrial membrane, but basically there is no cholesterol. That indeed you actually can permeabilize cells without permeabilizing the mitochondria because there is no cholesterol in mitochondria. This is then somehow again transported from the outside to the inside, to the inner mitochondrial membrane, used by, I don't remember what's the name of the enzyme,
CYP
five,
A1.
something
Yeah,
like
okay.
together with ferredoxin,
Sì.
yes, and then this hormone, that is the precursor,
Goes out. Yeah.
and you know, it's like magic happens, magic happens. It won't happen.
There are some like teams that are studying the star complex that should be involved in the movement of this precursor of the aldosterone and corticosterone. But to be honest, like when I was writing my thesis, there was not that many found in that field actually. So yes, it's not a really well described process. They say that the star complex should be a rate limiting step also, but for example, yes, it's for the import. But for example, in the treatment that we have done, we don't see it moving. You know. So actually, we are doing a lot of like at the transcriptomic level. So I would say that maybe we are not like assessing deep enough at the protein level what is going on. So maybe at the protein level we can see some differences in that. But yeah, there is not that much known actually.
All right.
Sorry, maybe I didn't understand. Your mouse model,
Yeah.
the
CYP11B sucre.
is the aldosterone synthase. Is that one? So AS in your plot. Or
Yeah, so CYP11B2 is the gene.
is the gene?
Yeah, and the enzyme is aldosterone synthase.
Okay, so when you do your KY
Yeah.
you have less aldosterone in this mouse model, right?
No, the aldosterone remain the same even in the Cre-Wild type and in the Cre-Cre. So there is no changes in the aldosterone production in the three genotypes, so wild type, wild type, and cre. But what we are observing is that there is a decrease in the, by Western blot analysis, of the aldosterone synthesis in male, only the homozygote cre, and in female we have wild type, and cre. Until reaching the cre, there is a significant decrease.
How is possible that aldosterone stay in the same level if you don't have the enzyme?
So that's what I said. So we have no studies that are showing the capacity of the enzyme to produce aldosterone. Okay, so as I show after for the immunofluorescence, it seems that there is a trend in increase of the percentage of cells that are aldosterone synthase positive. So our idea, I mean, I don't know if we can prove it, but that's what we think. So we have less total amount of enzyme, but there are more cells that are expressing a lower amount of this enzyme. So what we think is that maybe even though it's a lower amount, that is enough to sustain the production of aldosterone, but you know, that's something that is kind of difficult to prove. So even though we wanted to create a perfect mouse model with the bitsukre and everything, we didn't. What we think that happened is that, you know, that in the 3' untranslated region, you have some sequences that are stabilizing the messenger RNA. What we think is that maybe inserting the CRE and the IRES sequence in that part, maybe we went to touch one of those sequences, unfortunately, and that has the consequences of that.
Did you measure NADH levels?
So I did. I see no differences, but I mean the qPCR in that moment it was not perfect, so that's something that we wanted to keep for the review. So we already have everything ready, but I have to do it soon.
Yes.
So when you put in a low salt diet,
Yeah.
then you have like angiotensin, and you have more aldosterone.
Yeah.
Right. So and then you have a high transition from the zona glomerulosa to the fascicolata.
No. So it depends. In male mice, what we are observing is that because of the stimulus, there is a blockage of the conversion between zona glomerulosa and fascicolata, and a faster recruitment of progenitor cells to become zona glomerulosa.
So more in zona glomerulosa. But you think that this is dependent on aldosterone? So did you try to increase aldosterone without the low salt diet?
But if I do an injection of aldosterone, what is happening? I suppress the pathway, because I have higher aldosterone, and so I have more retention, and so the renin-angiotensin system goes down. So there are, for example, so to regulate the thickness of the layer, the main actor is the Wnt beta-catenin pathway. There are many studies that show, for example, overactivation of the gain of function mutation of the beta-catenin when you remove the regulatory subunit, leading to an overactivation. So it's always activated, and you have a thickness of the layer and also a penetrance of the zona glomerulosa into the, zona fascicolata part, let's say. And what we are observing is that most probably the Wnt beta-catenin pathway is controlling the expression of B2, but not the other way around. So there is a certain interconnection between these two pathways that regulate the zona glomerulosa identity, but it's not that clear, it's not direct. And we see that with this treatment, with 2 weeks, from 4 weeks to 6 weeks of the mice, is not enough to see a thickening of the layer. But for example, there are other studies where they were using adult mice, so well-developed mice, where they were performing a treatment, I think at least of 4 weeks. And in that case, you see increase of aldosterone, all the things that we are observing, but also a thickening of the layer. But not in our model. Not with this timing that we are doing. That's why I said in the perspective, I think that would be interesting to be able to modulate better, because this is first described at this time point, and also there is this huge limitation, because if we want to do some study of the transdifferentiation, as you saw, I didn't show you actually, so the main way to do that is to track the GFP progression. But when we reach 6 weeks, that is the first time point after the diet, we are around like 70%, 75% of the cortex that is already GFP positive, meaning that you are already reaching the lower part. So if you have a blockage between zona glomerulosa and fasciculata, it's already too late. You cannot see that. So we are trying to see if there is an increase in the tomato positive, because, so that's the only things that we can do. With an inducible one, you can start the diet, you induce the mice, you start the GFP, and you see if the GFP is retained there, or is going down, or what is going on. But I don't think that the lab is going to do that. So I don't know. This is going to be maybe someone else's project. I don't know.
It's just the sun diet, or some like sugar or something else that is causing?
So sugar, no, but as I said in the hypothesis, there is stress that can be a modulator of the other layers, so of the zona fasciculata. The zona fasciculata is controlled by the hypothalamus pituitary axis, so the stress leads to the release of corticotropin releasing hormone, release of ACTH by the pituitary gland, and after it reaches the zona fasciculata. In the zona fasciculata, after you have the stimulation in the production of cortisol in human and corticosterone in mice, and these lead to like more a thicker layer or a decreased layer. So what we have done, and I couldn't show you, but we have performed the dexamethasone treatment. Dexamethasone is an exogenous glucocorticoid that leads to the suppression of the HPA axis, and meaning leading to the atrophy of the gland, of the zona fasciculata layer. And what is interesting in those mice, we could observe a difference in transdifferentiation, because at the time point 6, the cortex, the controlled mice, they are like this. The treated mice, they are like this. So it's really shrinking a lot the organ. And what we see is that at 6 weeks, the GFP is 100% of the cortex, meaning that you have a faster recruitment of the zona glomerulosa. And also what is interesting is that in the spatial transcriptomic, we see on the other end that for a treatment that is like meant to target the zona fasciculata, there is a huge zona glomerulosa recruitment. So there are transcriptional changes in the zona glomerulosa that are not observed, for example, in low salt or high salt diet. That was really interesting. When we are doing a treatment for one layer, we see the other one responding.
What did you say?
Strong communication.
Yes, exactly.
So the overall question is, what's the mechanism?
Okay, so
How is it possible that cells migrate, transdifferentiate, die, and all this is controlled by external stimuli?
So what we did is the modulation of the two main pathways that regulate the production of the steroid hormones, that should be also correlated with an increase in the Wnt or the PKA pathway, that are the main pathway that are involved in the process of transdifferentiation. So we know that there is a mutual inhibition between these two pathways, but actually we are not observing these changes at the moment and in the study that we have done. So the process...
We're not observing the changes. Does it mean that you don't see the reciprocal influence?
So
or does it mean that you never tested it?
No, we have done it. For example, the Wnt beta-catenin for the high salt and low salt, we have analyzed them at the qPCR level. No changing, and that is also correlated with what we are observing, that there is not change in the thickness of the layer. So it's kind of confirmed, and that is fine. What we don't understand is what is going on in the dexamethasone treated mice, where we have a faster transdifferentiation, because in that case we have an activation of the Wnt beta-catenin pathway. That is the pathway of the zona glomerulosa layer, not of the zona fasciculata. So, again, treatment for one layer is involving the other one.
No, I get it. But I mean, I'm a cell. I'm at the glomerulosa.
Yeah.
Okay. And then somehow I migrate.
Yeah.
And while I migrate, I transdifferentiate.
Yeah.
Okay? So this means that most likely there is a gradient of a morphogen that is doing the job.
Mm-hmm.
Okay? What's the morphogen?
That's a good question. That's what like.
Why the kind of stress?
Because it is like going inside,
So actually, we have done light sheet microscopy on cleared organ. So with the tissue clearing, we could scan all the cortex and also all the medulla. Not perfect. We don't have perfect data, but what we observe is that showing all the different layers with the 3D reconstruction of the organ, we see that the I don't know if I show you. You can see in the transdifferentiation that there are some spot like you have the cortex. You have the cortex, GFP positive cells, and after. Okay, now it's too much. Okay, you can see here, for example, that you have this like spot of cells GFP positive that seems that they are alone. Okay, what we have observed with the 3D reconstruction.
What do you mean?
So you see that what you would expect is that you have a kind of this process that is just progressing with the GFP cells that are entering, but I would not expect to see like a spot of GFP positive cells surrounded by of tomato positive cells surrounded by GFP positive cells.
I think that this is because you are flattening a 3D process onto a 2D.
Exactly, but in the 3D, what we have seen is that those spots, let's call it spot, of tomato positive cells, in reality they are column of tomato positive cells.
Like this. So
they go from the top.
Take a slice.
Yeah.
Okay, and so you're seeing the bottom of
Yeah.
tomato positive cells, but in reality this comes from here. Okay, and that's a common issue in microscopy.
No, I'm not saying this. So in the 3D, what we see is that, for example, when you start to enter in the zona glomerulosa, okay, you see that maybe in the zona fasciculata, you pass all the zona glomerulosa that has to be GFP positive because we are seeing it at 6 weeks. And after we are in the zona fasciculata, we start to see the first group of tomato positive cells. And entering until reaching the medulla, we see that those cells, they continue until the medulla. So they're all interconnected, the tomato positive cells. And in this case, we are just cutting most probably the column. We are cutting the column that is like this in a 2D. So that is interesting because there is a group of stem cells that you can see here, that are not at all described actually in the literature. There's only been identified that are those that are kind of like neuronal cells, nesting positive cells that has this filament that go along the cortex, and I think that they must be involved in this process or in retaining the tomato positive cells and recruiting the progenitor in the zona fasciculata directly, or in some way.
This doesn't answer the question.
No, I know, but there is no answer to the question. I actually don't know. My idea is that, or there is I think that the mitochondria they have a really huge involvement.
I don't think it's mitochondria, but there should be a.
No, but I think that.
What do you think? I mean, maybe the nesting positive progenitor cells, they are secreting something.
Mm-hmm.
Okay, and because of how they are organized, they might generate. Or maybe it's the medulla.
No, there is no connection with the medulla.
I know, but this is not a physical connection. It's a chemical connection. Okay?
Yeah.
Did people try to actually see whether this happens also when you ablate the medulla?
So from the medulla side, no. But modulating some of the actors involved in the identity of the cells and everything, it has been shown that there is a misplace of the medulla. So, during the embryo development.
Yeah, so the point is that people never actually ablated in the adult animal the medulla surgically, okay? And checked whether, there was a issue in the transdifferentiation.
So, there are experiments of enucleation. So the enucleation is the removal of the entire organ, leaving only the capsule and the progenitor cells. And what has been shown, I think that it has been done in rats, for like size reason also, show that there is a total reconstitution of the cortex, but not of the medulla. Yes.
Okay. Which leaves the question open.
Yes, which leaves the question open that there are many teams working on that. There has been like many modulation of the process, but never like a description of, for example, physiological response to different treatment like this for this process. And I think that would be perfect to like cross this.
No, what are the surface receptors that you identify in your glomerulosa and fasciculata cells that are different? I would start from that because you have the data set and you might actually have, you might be able to pinpoint what is the morphogen that causes the transdifferentiation. Because there should be a gradient of the strain.
Yeah.
Because it probably goes up in the
Glomerulosa.
interfacial region, okay, and then goes down in the when you transdifferentiate. And so you might be able actually to. This should be something that is outside, okay.
Yeah.
So maybe you can find it by using this strategy. Not so complicated.
Yeah, that can be an idea. So I know that, for example, the like main receptor, for example, for the stimulation of the production of the hormones, there is only the zona fasciculata that has a difference because it doesn't express the AT1R receptor. But you know, in the tissue is really heterogeneous. So it's not that easy as you're saying that you see a gradient because the component of the capillaries that are going along all the cortex is really thick. So you have a huge heterogeneity of the different cells that are present in the tissue. So, but that can be an idea, of course.
But also that it's super good that you switch first from the signaling pathway with and beta-catenin and then cyclic MP and PKE. Then I don't know whether it is the configuration like this, but it seems like that the fasciculata, the cell are more tight together. And
Yeah, they're different. So so this has been described in mice and not been really proven in human. So the zona glomerulosa in mice is disposed in rosette. So the rosette is this composition that it can go from 7, 8 cells until 15, I think, that they have kind of an apical part. And this is important for the propagation of the calcium signaling pathway between the cells. So when one is stimulated, it kind of connects all the others in the production of aldosterone. And they are characterized by extracellular matrix components that are present, for example, in the external part and not in the between the cells. And after when, and also the vasculature of that part is kind of round shaped like this.
but it could be that you have after in the fasciculata the increase in the cyclic MP and PKE pathway is because the cell there are more near, maybe also you have less vessels, and so the cell feel like a starvation.
And you have more vessels in the fasciculata.
They have more
Yes, because for example in the fasciculata, the vessels they are like they are really like the ray of a wheel, you know. They are exactly like this until getting to the medulla. And the composition of it is really, really thick, and you can see differences when you treat, for example, with the dexamethasone, you see that the vessels are disappearing because it's going to atrophy the layer. And the disposition and also the size of the cells, they are exactly like this. So it's more ordinated in zona fasciculata, they are like bigger the cells than the one in the zona glomerulosa. And in the X zone, they are exactly like this. So they are impossible to quantify more or less because they are that small and that dense. No, it's not inducible, unfortunately.
When you talk about transition, that you are already not tracing all of the cell, right? So have you ever tried something kind of like, I don't know, even EDU or you're in one pulse of the one moment and then there is labeling of the cell to see the transition of the cell?
Translate
You mean
So he was asking whether you want to do, you planned, ever planned to do a pulse chase experiment with BRDU to look at whether there are changes in proliferation.
Because it's now for the moment we just see the expression associated.
Mm-hmm.
When you say transition, maybe you mean this.
So we haven't planned to do this experiment. At first, what we tried to do, it was the window strategy. So to place the window of in the side of the, like in order to be able to scan the tissue live timing. So every day to see which cells they're entering. But to be sure, I don't know. No, we haven't think about it. An idea that we had at one moment is using the confetti mice instead of the MTMZ. So the confetti mice, they have a different wavelength of expression. So for example, you have one cell that is violet, another one that is GFP, one another that is tomato, and you can track each cell. So because all the other cells that are originated from that will be of the same fluorescence, and so you should see from one originating after the others expanding like this. But we didn't do it at the end. But the idea was also to try to, for example, isolate them with the fax and sort it with the fax in order to be able to isolate and do the single cell with those. But actually sorting them by the GFP, the tomato così, when you do the digestion of the tissue, you lost everything. I mean, there is one, there is one team that managed to do that, but I asked many times the protocol, I have no response. So actually I don't know how they did, because at the fax I was able to see GFP cells, tomato cells, and also co-positive cells, meaning that those are the cells that are stopping the expression, that are expressing the GFP starting, but they didn't get rid of all the others still tomato positive, okay? But after, when I went to the sorting, I saw a lot, like 90% of double negative cells, so nothing. And at that moment I said, okay.
Okay. Any other question? No? Thank you very much.
You're welcome.